preventing septic implant failures in osteoporotic hip fractures in rat. So let me start with a brief overview of osteoporosis. It's a disease that weakens the bone and enhances the risk for sudden fractures and bone fractures as shown in this video. So what happens in osteoporosis is there is a destruction of the bone homeostasis and there is increase in bone resorption that takes place because of the enhanced activity of osteoclasts, and World Health Organization has given a criteria for diagnosing this osteoporosis, and it has given that based upon the bone mineral density of the bone and the t-score calculated from it, the t-score range of minus 2.5 or less is considered for a patient to be osteoporotic. So what happens is that whenever such type of fractures occur in an osteoporotic patient, it is fixed using several implants which includes K-wires, screws, dynamic hip screws, etc. And this situation worsens whenever there is an infection in these implants as well in the osteoporotic patients. So the major causes of these infections include open fractures or any type of surgery or secondary surgeries. The major bacteria involved in this osteomyelitis type situation is Staphylococcus aureus. So this is the infection that occurs in these type of bones, and it is considered one of the most serious complications in orthopedic surgeries. And there is a requirement to enhance the strength of the bone so that the secondary implants that will be implanted do not fail again. So there are very limited strategies that are available to treat such type of issues. So we have designed a study where we have designed the structure like that. We made an implant associated osteomyelitis model in an osteoporotic rat where we utilized nanocement-based carriers for delivering several bioactive factors and antibiotics at the site of fracture so that we can achieve bone remodeling, infection control, and improved mechanical strength. So in the first phase of the study of this experiment, bilateral ovarectomy was carried out in which BMD T-score was calculated, and as per this formula, different regions of interest were taken which were supposed to be affected by osteoporosis, and we have seen a significant decrease in the T-score values in all of these region of interest in a period of 3 months, which was in the range of -4, which is the range that was provided by WHO. And in the second phase of this experiment, we created a fracture in the femur head canal, and then we have infected it using S. aureus mediated K-wire implant. And then in a period of 14 days, this is the DEXA image of the same, and these are the harvested K-wires in a period of 14 days. So what we observed during this time of 14 days, there is a significant decrease in the weight of the animals, and TLC and DLC values of the animals has significantly increased. So in our treatment strategy, we have utilized nanocement as a carrier for different type of bioactive factors, which was shown here in 3 animal groups, which includes the combination of MSC exosomes and zoledronic acid. In this group 4, we can see it is a combination of BMP and zoledronic acid, whereas in the group 5, we have utilized the combination of all these bioactive factors in addition to rifampicin as antibiotic. So what we observed after a period of 4 months using DEXA analysis, that there is a significant increase in the bone mineral density, and the T-score values of the treatment groups has reached the normal range. These are the micro-CT images where we can see enhanced bone remodeling, and the micro-CT parameters we can see in the treatment groups. Bone volume by tissue volume percentage has significantly improved in the treatment groups. And if we see a trabecular number, it has also significantly increased, and there is a decrease in trabecular separation, showing that good bone formation has taken place in the site of the defect. So also, as I mentioned, the mechanical properties are very essential. So what we see is that the peak fracture force that is required to break these bones after the treatment has significantly increased so that the secondary fractures can be avoided. The bacterial load analysis done for the ex vivo samples isolated from the rats shows that in the treatment groups we are not able to find the bacterial colonies as well. The histological analysis shows a significant improvement in cell infiltration and collagen deposition in the treatment groups, where we are able to see in these treatment groups cells has infiltrated in the fraction site and collagen deposition has taken place in the fracture area. So we can conclude from this study that successful bone regeneration and infection control were achieved, and with these bioactive molecules and antibiotics which were loaded in the nanocement. And these MSC exosomes, they are showing a potential alternative for BMP2 in bone regeneration when used in combination with zoledronic acid. I would like to acknowledge my thesis supervisor, Professor Ashok Kumar, BSB faculty and staff, Animal Facility IIT Kanpur, DBT for my PhD fellowship, and all previous and current lab members. Thank you very much.
Thank you, Aman. Our next speaker is Rafat Ali.
Good evening to all. My title of talk is Hydrogen Sulfide Releasing Peptide as a Multifaceted Approach to Alzheimer Disease. So, as you know, Alzheimer disease is a progressive neurodegenerative disorder in which humans lost their thinking ability. I will explain how works the Alzheimer disease by taking the famous example of William Otermullen, who was diagnosed with Alzheimer disease. The first picture he was painted before he was diagnosed with Alzheimer disease, and later pictures he was painted after the diagnosis of Alzheimer disease. So you can see that painter was not able to draw his self portrait. So you can imagine how badly the Alzheimer disease affected the human thinking ability. So if you look at the factor, Alzheimer disease is a multifactorial disease, and several factors like oxidative stress, protein aggregation, degeneration of cholinergic neurons, and so on leads to the progression of Alzheimer disease. So, due to multifactorial nature, Alzheimer is generally considered an incurable disease. So, we need a molecule which will target multiple aspects of Alzheimer disease. So, in this context, hydrogen sulfide will emerge as an important player, and in human brain it is shown to display significant neuroprotective activity, such as it will increase glutathione level and it scavenge radical species. And it was observed that in Alzheimer disease patients have low level of hydrogen sulfide in their brain, and it is very difficult to deliver hydrogen sulfide from outside due to its gaseous nature. So several inorganic salt and small organic molecules were developed by the scientists to tackle this to deliver this hydrogen sulfide from exogenous source. But from these molecule it is not possible to deliver hydrogen sulfide in the controlled manner. So we come up with the strategy in which we design and synthesize hydrogen sulfide releasing molecule, hydrogen sulfide releasing actually peptide conjugate 1 and 2, which will form by combining this H2S releasing molecule with these anti-aggregating peptides. These peptides will form self-assembled structure in aqueous solution, and due to self-assembly it will releases hydrogen sulfide slowly and in sustained manner. So as I have told that deposition of A beta in the brain is the main culprit of Alzheimer disease. So we evaluate our peptides for their ability to inhibit A beta aggregation. For that we perform this thioflavin T fluorescence assay, and in this assay we found that our peptide conjugate was significantly reduced the A beta aggregates. This was further confirmed by this atomic force microscopy imaging. If you see the first column, you can see that after 24 hours of incubation, the A beta start to form this fibril like structure, whereas in presence of peptide conjugate no fibrils were seen. For in vivo system we use this C. elegans form. This C. elegans have cytochrome P450 and NADPH enzyme, which allows in vivo release from these type of molecule, and with the help of this coumarin we can quantify this hydrogen sulfide release inside the C. elegans, because in presence of hydrogen sulfide this molecule give green color fluorescent molecule. So if you look at the fluorescent micrograph, you can see that these conjugate 1 and conjugate 2 treated C. elegans form have show more green color fluorescent, which correspond to more hydrogen sulfide inside C. elegans. For A beta aggregation we use this transgenic C. elegans form, which will express human A beta protein in their muscle. If you look at this untreated form, this white deposit or blue deposit is nothing but A beta aggregates, and when we treat these C. elegans with our conjugate, there is a significant reduction of A beta aggregate was observed. So moreover these peptide conjugate were significantly reduced this reactive oxygen species generated by hydrogen peroxide inside C. elegans. So actually what happened, these A beta aggregate and reactive oxygen species mainly degenerate the cholinergic neuron, which will result in the low secretion of this important neurotransmitter acetylcholine. So we found that our peptide conjugate was significantly increases the acetylcholine in transgenic C. elegans form of C. elegans model of Alzheimer disease, and we conclude that decreased oxidative stress and A beta aggregation by our peptide conjugate is mainly responsible for this increase of acetylcholine. So in conjugation we have developed hydrogen sulfide releasing peptide conjugate, which will target three important factor of Alzheimer disease, and we observe significant reduction in this A beta aggregate and this reactive oxygen species, as well as we observe significant increase in acetylcholine level was observed in C. elegans. So our future plan is to test these peptide in mice model of Alzheimer disease with the hope to recover this neuronal loss caused by Alzheimer disease. So finally, I would like to acknowledge my mentor Professor Sandeep Verma and all the collaborators involved in this work funding agency, and thank you for listening.
Thank you, Rafat. Our next speaker is Preeti Sati.
Good evening, everyone. This is Preeti Sati, and today I'm going to talk about my research topic, membrane-free liver on a chip for drug screening purpose. So, first of all, testing experimental drugs with human first is very risky and challenging process. Also, drug discovery with the conventional platforms and with the animal models are not that effective, time consuming and expensive processes. So, to overcome these challenges, microfluidics and tissue engineering are working towards to develop novel organ-on-a-chip platform for drug screening purpose. Inspired by organ-on-a-chip platform, liver-on-a-chip can be the better alternatives to the animal models and the conventional models, because liver plays the main important role in drug metabolism. So closely if we closely look at the structure of this liver, so we can see that liver is made up of the thousand of the lobules, and in each lobule, because liver is made up of the 90% of the hepatocyte cells, so hepatocyte cells in the liver are arranged in the hepatic cords, 3D hepatic cords, and separated by the liver sinusoid, which are the vascular channels, and which the endothelial monolayer cells are flanked. So these hepatocyte cells are separated by the endothelial cells, and they are not directly exposed to blood flow. In fact, they fed by the nutrients and media via diffusion. So to mimic such kind of complex architecture, or we can say to mimic such kind of two tissue interface in liver on a chip platform, previously people have people have utilized these microporous membranes and micropillars inside the channels. So these microporous membranes are, we can see that, separating the two cell types, and we can see that these microporous membrane inside in the bottom channel, they are protecting the hepatocyte cells from the shear stress of flow, and also from the pores of this microporous membrane the mass transfer is from there. Similarly, in the micropillar based chip, between the spaces of these micropillars, the flow can be introduced, and that's how they are protecting these hepatocyte cells from the shear stress of flow. So, but these models have the limitations that the membrane cannot completely remove the convection on the hepatocytes. Also, these kind of model are constructing the 2D platform, because if we can see that these hepatocyte cells in the bottom channel, they are growing in 2 dimension, and they are actually fails to mimic tissue structure and functions, because they are not properly getting the native 3D microenvironment. So to overcome these challenges, we have designed a membrane-free single-channel membrane-free liver on a chip, where we have utilized the PEGDA hydrogel as an alternative to the membrane, which is overcoming all the challenges of the membrane. So this PEGDA hydrogel is also modified with the CGRGGS peptide, which are the cell adhesive peptide. And first of all, we have encapsulated the Hep2 liver cancer cells and fibroblasts inside the hydrogel, and then we can see that these PGDA hydrogel not only protecting the hepatocyte cells from the shear stress of flow, but also the mass transfer was there, and via diffusion the Hep2 cells are getting the nutrients and the media. To achieve our final step, we have done the peptide immobilization over the surface of this cell encapsulated PEGDA, and that's how after 24 hours of seeding HUVEC cells, we can see that with the help of self-assembly, these cells are forming the monolayer. So that's how we have achieved our membrane on a chip platform. This model is actually can also be act as a 3D liver tissue chip. So to prove our model, we have done some FDA stain, live staining of the encapsulated cells and the cells over the hydrogel. So we can see that these Hep2 cells inside the hydrogel are with the passing day, they are proliferating inside the hydrogel, and also these HUVEC cells also covering the surface of the hydrogel. Well, because this model give the functions and viability up to 15 days, so we have tested the live date analysis up to for 15 days. So we can see at each passing days, these cells are not only encapsulated cells, but also the surface cells are covering the full hydrogel. We have also tracked these monolayers with the green tracker, and these encapsulated cells with the red tracker. So we clearly can see the difference, that how these HUVEC monolayer are covering the upper surface of the hydrogel, and the red cells are showing the encapsulated cells encapsulated inside the hydrogel. So now finally we have tested the liver specific functions, urea secretion and albumin secretion. So we can see that we have observed that this model is giving functions and viability up to 15 days, and the maximum function is up to 9th day, but in the 15 day itself the function was more than the first day. Similarly, the thing we have observed in the albumin secretion. Now we have tested the sensitivity of the model with the hepatotoxic drug, 10 millimolar hepatotoxic drug. So we can see that this drug is toxic for the liver cells. So liver cells are dying at each passing day. We can see with the red color images of red color are the dead cells, and green colors are the live cells. With urea analysis of 10 millimolar this acetaminophen can show that the urea secretion has became half. We have done the comparison without drug and with the drug. So first, for the first time, we have developed this membrane-free liver on a chip platform, which not only overcoming the challenges of the membrane, this can also act as a 3D liver tissue chip. So this PGDA hydrogel is not only providing the 3D microenvironment to the cells, but also protecting the cells from the shear stress of flow. So I would like to thank my thesis supervisor, Professor Shri Shiva Kumar, and our collaborator, Professor Sandeep Verma and group. And I would like to thank my lab mates and my seniors. Finally, I would like to thank IIT Kanpur for all the funding and support. Thank you so much.
Thank you. So with that, we move on to the last speaker for today. Do we have him here? Okay, here. Okay. So she's the last speaker. Go ahead. Yeah. She's a BTech student. She's an undergraduate student.
So, good afternoon everyone. I am Riddhima Chaudhary, and today I will be presenting my MyTex internship that I did in last summer. So I did this project under Chantal Guillemette in her pharmacogenomics lab, and it was in University Laval, Quebec City, Canada. So the topic for my internship was to identify potential substrate of UGT2B17 and UGT2B28 enzymes. So let us look at what are UGT enzymes. So UGT stands for uridine diphosphate glycoside transferase enzymes. So they catalyzes the glucuronidation reaction, in which they add the sugar molecule from UDP sugar donor to variety of functional groups, that are carboxylic, amine group, and hydroxyl group, and endogenous toxic metabolites. They ultimately forms glucuronides that are inactive and water soluble. So they perform a special function in human body elimination process, and they maintain cellular homeostats, that they maintain the concentration of metabolites in our human body, and they detoxify the xenobiotics and clearance of used drugs. So even though they are well known for their process in elimination process, but there are very less systematic and cell metabolome studies that are available, and many of these UGT enzymes do not have any specific substrates known. So the objective of my project was to analyze the mass spec data, systematic and cell metabolome, and find out some specific substrate for UGT2B17 and UGT2B28 enzymes. So the methodology that I followed was, I analyzed the mass spec data from 3 different data sets. First one was from cell lines. So in cell line we had MEC1 cell line that is specific for mantle cell lymphoma, and GBM2 cell line, which is known for CLL disease, and LN-CAP cell line, which is a prostate cancer cell line. So MEC1 and GBM2 cell line are known for their overexpression in UGT2B17, whereas LN-CAP cell line is known for their overexpression of UGT2B28. And the second data set that I had was from plasma of prostate cancer patient, and there was 2 types of data. First one having germline deletion of UGT2B17, and the other one had the germline deletion of UGT2B28. The third data set was from plasma of Canadian longitudinal study on aging. So this data set was from cohort of Canadian population that were healthy, and they voluntarily give their plasma for scientific research and their bodily checkup. So in this data set there was knockout of UGT2B17 and UGT2B28. So the whole mass spec metabolomic data was of 2 types: N-targeted data set, that in which no metabolites are targeted, and another data set was from lipidomic, where specific lipid molecules were targeted. So to search for potential substrate of UGT2B17, I analyzed data set from GBM2 cell line and MEC1 cell line, which are known for their overexpression of UGT2B17, and in this data set I looked for metabolites that were downregulated. And similarly, from the plasma of prostate cancer, which was having knockout of UGT2B17, I looked for metabolites that were upregulated. In this way I came up with a shortlisted list of potential substrate for UGT2B17, and from literature review I collected more evidences that supports that they needs to be eliminated from our body. So further I went for performing glucuronidation reaction, and then by mass spectrometry it will confirm whether they are substrate of UGT2B17 or not. So in this way, the one metabolite that I found out was quinolinate. And similarly, I looked for potential substrate of UGT2B28. I looked for metabolites that were downregulated in LN-CAP cell line, and from this shortlisted list, I looked for metabolites that were upregulated in the plasma of prostate cancer patient and CLSA, which were having knockout of UGT2B28. And similarly, I made the list of potential substrate for UGT2B28, and evidences were collected from literature that supports that these could be the potential substrate of UGT2B28. Mass spec was done that give us the result whether they are potential substrate or not. So in this way, I came up with N-acetyl neuraminate as the potential substrate of UGT2B28. So this is the design, experimental design for enzymatic assay, and it was done to check whether glucuronidation is happening in the by these enzymes on these substrate or not. So in the master mix, microsomes from liver, kidney, and intestine was added, and quinolinate and N-acetylneuraminate were added, and one positive control was made. So DHT is known that it is that is the substrate of UGT2B28, which is UGT2B17, which is found in liver microsomes, whereas 3 negative control were made, and all were kept for overnight incubation, and then methanol was added to stop the reaction, and then mass spectrometry was done. Similarly, I found out these lipid molecules as the potential substrate of UGT2B17, and like sphingolipid and some lipids from LPC ester and LPE ester were found out as the potential substrate of UGT2B17. And for UGT2B28, similarly, PE ester and N-acetylglutamate and spermine were found out as the potential substrate. So in conclusion, I would like to say I found out quinolinate and N-acetylneuraminate as the potential substrate for UGT2B17 and UGT2B28 as their potential substrate respectively. And I found out 6 lipid molecule and 1 lipid molecule as a potential substrate for UGT2B17 and UGT2B28. And the future prospect for this would be that for the first time a potential substrate was founded for UGT2B28, and this will allow us to check the enzymatic assay for UGT2B17 and 28 enzymes, and it will also provide us an indirect way to confirm or to validate the overexpression of UGT2B28 in prostate cancer and bladder cancer cell lines. So at the end, I would like to thank MITEX program, the professor that supported me for this internship, and thank you so much.
Thank you, Ridhima. So with this, we come to the end of this last session, and I want to thank all the speakers, Dr. Shamik Sen, Dr. Jayendran Rao, Aman Nikhit, Rafat Ali, Preeti Sati, and Ridhima. So please join me in thanking all these speakers. So we have actually gained some time in this session, but I was informed that yesterday there was some shortage of time for discussion on abstracts. So we will close this session right now and start the tea session as well as the abstract session parallelly right now, right after this. So we break for tea right now. And you want to, she has a small announcement regarding NGO that her father runs.
Good afternoon, everyone. I'm Chitral, and I'm part of this NGO, which is a wildlife and environment protection NGO called the Junglies. And we have recently launched this anti-plastic campaign. So we are collecting signatures from educational institutes across the country. And I request you all to please take out your precious time and sign this petition, which will be given to the Prime Minister. And I'll be really grateful if you can support me in this noble cause. Thank you. So these forms will be kept at the registration desk. So if you could just sign with your names, and yeah, that'd be great. Thank you so much.
So we now break for tea, and all students who have their posters to be presented, please go ahead first, put up your poster, and then pick up a cup of tea.